Great. There we go. Okay, so it is my pleasure today to introduce Dr. Edgar Medina, who is joining us from the University of Massachusetts Amherst, where he is a postdoctoral fellow. So Edgar is an evolutionary cell biologist, as we're going to hear about, dissecting the origins of fungal organization and architecture, hence all these beautiful videos that we see screaming in the background. And he's working on Chytrid fungi, which, if I'm not mistaken, I think will be the first of this seminar series that we run to focus on Chytrids, which is awesome. So kind of a whole new branch of fungi. Edgar was born and raised in Colombia, where he obtained his bachelor's in science in biology and a bachelor's in science in microbiology. Am I getting that right? Two double?
Yep.
Okay, great, amazing.
Yep.
At the Universidad de los Andes. He holds a PhD in genetics and genomics from Duke University, where his work focused on the evolution of fungal cell cycle control and developing genetically tractable model, or the first genetically tractable Chytrid model. So currently, Edgar is an HHMI Hannah Gray postdoctoral fellow, which is a very prestigious postdoctoral fellowship in the laboratory of Dr. Lillian Fritz-Lehlen at the University of Massachusetts Amherst. And so we are delighted that he is here today to share some of his research on Chytrid fungi. And we'll let you take it away, Edgar.
Thank you so much, Rebecca. Thank you so much for the chance to share with you all some of my work on the evolution of Chytrids and fungal developmental programs. Now, I think we are all fascinated by the organization of the fungal kingdom. We have these beautiful and elegant shapes and forms. And what is fascinating to me is that fungi and animals share a common ancestor about 1,200 million years ago. And since then, they have diverged drastically in their developmental programs, organization, architecture. And this is despite sharing a very similar core cell biology, which has allowed model fungi like yeast to all those findings to be translatable to human health. Now, most of the organization of a eukaryotic cell depends on the cytoskeleton. And this is a beautiful poster made by Professor Katrina Vell when she was in the lab. And you can see in green the actin cytoskeleton and in teal the microtubules. You can see the diversity of shape and form of the cytoskeleton is reflected in the diversity of shape and form of the eukaryotic cell. So if we want to understand the organization of developmental programs, understanding how the cytoskeleton regulate those shapes is a fundamental task. And I will focus in the two of the main elements of the cytoskeleton, the actin and the microtubules. And actin is fundamental for the supporting and the deformation of membranes in many biological processes. So you have cell crawling, for example, on the left, you see this Chytrid zoospore moving and crawling, and actin is fundamental for that process. You also have endocytosis and exocytosis and intracellular trafficking. Actin is fundamental to it. And in the lineages of the fungi, the animals, and the slime molds, contractile actin is used for the assembly of the cytokinetic ring, which allows cytokinesis in these lineages. In the case of microtubules, you have the assembly of the mitotic spindle. Microtubules are used for the movement of the organelles inside the cell and the assembly of the motile cilium. Now, if we want to understand the evolution of the developmental programs in fungi, we need to focus in the deepest lineages within fungi, because this allows us to understand across the whole kingdom what are conserved traits specific of the lineage, and on the other hand, traits that are shared with animals and thus likely ancestral to the fungi. Now, Chytrids are special within the fungi because they are the only ones that share with animals the capacity to produce cells without cell wall that can crawl on surfaces and swim with a motile cilium, and this is called the zoospore. Now, the general life cycle of Chytrid fungi oscillates between this ancestral type of cell, the zoospore, and a more typical fungal developmental program. So in the generalized life cycle, you see the zoospore, it finds the proper niche and it retracts its cilium and builds a cell wall. Then it germinates with polarized hyphal-like structures that are called the rhizoids for attachment and feeding purposes. And meanwhile, the main body of the cell, which is called the sporangium, grows in size. At the same time, the nuclei divide synchronously without cytokinesis, forming a sinusoid, like most fungal cells. What's interesting is that at the end of the life cycle, all the content of the sporangium is allocated into individual uninucleated cells without a cell wall that are the future zoospores. And around each nucleus, a new cilium is formed. And the zoospores have then undergo abscission, cell separation, and they're released into the environment again. So this oscillation between ancestral traits and typical fungal developmental program makes of Chytrids a very good model system to try to understand the emergence of these fungal traits and the disappearance of the ancestral ones. Now, what is fascinating about the cellularization process, and here you see a three-dimensional reconstruction of a point scanner image of a Chytrid Specialomyces, is that you see that the whole mother cell is filled with nuclei, and you see the color, the relative position across the nuclei, and around each nucleus, here you see peak cellularization, and in green you see the membrane. You have a polyhedral membrane that it's assembled around each nucleus, and all the nucleus are packed in this beautiful three-dimensional lattice. And if you give this cell to an animal person, you don't tell them anything, they're going to think, actually, this is an animal embryo. What is fascinating about this process is that animals build this kind of tightly packed multicellular structure out of usually consecutive nuclear cellular and nuclear divisions. And they have this packed organization later on. What is fascinating about Chytrids is that they don't have to go in through successive cytokinetic processes, but they build this beautiful lattice out of scratch from a sinusoid within 1 hour. So my main interest is trying to understand how do you assemble this beautiful feat of geometric self-assembly from scratch using the cytoskeleton. Now, the three-dimensional nature of this process is important because we know a lot about conventional cellularization. This is conventional cytokinesis, in which the nuclei, the two daughter nuclei of a cell, are aligned in a single line. We know a lot from animal model systems and budding and fission yeast. So you have a 1D organization of the nucleus, a single line. We also know a lot about two-dimensional cellularization processes in which the nuclei are arranged in a single plane. And this is the case of the base model system on cellularization that we know of, Drosophila syncytial cellularization, in which the nuclei migrate to the plasma membrane, and then they're arranged in a single plane. And all those nuclei cellularize, and the product is the epithelium, which is what is used for the morphogenetic programs of Drosophila. We know a lot about it. Now, we don't know much about three-dimensional cellularization, in which some nuclei are surrounded all around by other nuclei, and all of them undergo cellularization. So you have to build a cytokinetic furrow on many different planes and at different angles. Now, we have no model system until now within the pestacons of a three-dimensional cellularization program. So we cannot assume that all the principles that we know from one-dimensional and two-dimensional cellularizations apply to solve the problem of 3 dimensions. So that was my task, to try to understand what are the general principles of three-dimensional cellularization using Chytrids. And I'll focus on 3 main steps in the cellularization process. First is how nuclei find their position and their dynamics in the cellularization process. Second, how the Chytrid cell chooses the places in which new furrows are going to be formed in 3 dimensions. And 3, how do they actually assemble this 3-dimensional lattice? I'll start with nuclear positioning. So what you're seeing here is a Chytrid cell with a fluorescently tagged nucleus, and in magenta, you see the membrane. And when I'm going to play the video, they're going to start undergoing cellularization, and I want you to focus on how the nuclei are moving. You're going to see they're really dynamic and stochastic in their apparent movements, but hopefully right before they enter cellularization, you're going to see a very brief period of time in which the nuclei migrate towards the brain. And I'll show the video multiple times so you can see the dynamic of the process. So they move around, they're assembling the lattice. Once it's assembled there, you see cell separation, and each zoospores becomes individual, and then they escape from the mother cell through a hole. So what you were able, hopefully, to see is the last nuclear division. Then they migrate to the plasma membrane. They moved as they cellularize very stochastically. They separate, and they get released. So we can see it one more time. So, and all this process takes between 1 to 2 hours. So these are still images of this process, so it's a little bit more easier to digest. So 20 minutes before the entry into cellularization, you have nuclei that are more or less homogeneously distributed. And as you start or approach cellularization, you see the migration of many of that nuclei towards the plasma membrane, and that coincides with the start of the emergence of these membrane planes towards that project towards the inside of the cytoplasm. To analyze in more detail this kind of process, what I did is that I got every sporangium and I then fit an ellipse in the sporangium, 2 ellipses, one in the outside, one in the inside, and this generates 2 shapes, the outside donut and then inside ellipse, both of them with equivalent surface area, about 50% of the surface area. And what we can measure is the number of nuclei in the outside versus the inside. And that ratio can tell us what is the bias in the distribution of the nuclei. If the ratio is more than 1, it means that there's a bias towards the outside in the sporangium. And if it is 1, suggests a homogeneous distribution in the number of nuclei outside and inside. And what we can do is track this ratio across the 40 minutes before entering to cellularization and 40 minutes after entering to cellularization. And what you see, there is a very brief period of time of about 10 minutes in which there is almost doubling in the number of nuclei outside versus the inside. And once that brief period of time passes, again, the nuclei become homogeneously distributed in the sporangium. So there is a very narrow window of time in which the nuclei migrate to the plasma membrane and they come back. And we think that this is an important trigger in the initiation of the cellularization process. So we have that nuclei are homogeneously distributed, they migrate to the plasma membrane, and they come back again towards the inside. So is this related to the selection of the division site in Chytrids? So we watch more carefully that kind of images that I showed you before and track what was happening with the nuclei as they approach the plasma membrane. And what we found is that in the proximity of the nuclei when they approach the plasma membrane, we see the formation of some vesicles of membrane. And with each nucleus that we see a vesicle in its proximity. So we wonder if there was actually the location of the centrosome. So chytrids, compared like animals, have a centriolar centrosome, and that centriole becomes also the basal body of the cilium that is synthesized inside the sporangium. And what is interesting is that during Drosophila cellularization, you have the same migration of the nuclei towards the plasma membrane, and each nucleus has its centrosome facing towards the plasma membrane. And it has this cage of microtubules around it and an inverted basket. And the interaction between the microtubules of adjacent nuclei is what is important for defining this place of the cytokinetic furrow in two-dimensional cellularization. So we were wondering if this was also the case in the chytrids when they migrate to the plasma membrane, if the centriole is facing towards the membrane, and if this is important to define where those vesicles that initiate cellularization are happening. So to test this, I generated a strain with a fluorescent tag component of the centriole. This is SAS6. And now each centriole is going to be colored in green. What we found is that when these green dots that are the centrosomes are approaching the plasma membrane, we see the formation of that vesicle in the proximity of the green dot. As we progress into cellularization, we see that that vesicle turns into that furrow that invaginates and extends towards the inside of the sporangium. What was surprising to see is that the green dot gets dragged with the furrow as it elongates towards the inside of the sporangium. So that was a little bit hard to see with the SAS6 because they're so small and they come out in and out of focus in live cell imaging. So we turn into a strain that has a fluorescently tagged alpha tubulin. And on the right, you see a video, which is the frame at the middle of the video that I'm going to play to you. And each of those arrows are showing the location of the interaction of 4 different nuclei with the plasma membrane. 3 of those nuclei have already undergone the last nuclear division, and there's 1 nucleus that still has 2 centrosomes, and that is going to undergo the last nuclear division. Up to this point, we thought that they were completely synchronized in their nuclear divisions, but it turns out some nuclei are not completely synchronous in the last nuclear division before cellularization. But hopefully you will see in the negative space, the nucleus, and in the green dots, the centrosomes as they interact with the plasma membrane, and hopefully you'll see what happens with that interaction. So the nuclei approach the plasma membrane, they start interacting with it, and you form those vesicles of membrane, and afterwards they get pulled inwards, and that pulling inwards drags the nucleus with it. So it seems that the nuclei tether to those membranes as they get dragged inwards. I'll play it one more time so you can see it. So what we think is happening is that the nucleus migrates towards the plasma membrane. There's the formation of that vesicle, and the nucleus tethers through their centrosome into that vesicle. And then this furrow elongates and extends towards the inside of the cell, and the nucleus gets dragged with it. Now, as I introduced you before, during two-dimensional Drosophila cellularization in animals, they have a similar architecture with the centrosome facing the plasma membrane, asters of microtubules around it, and in the case of Drosophila, is the interaction between the asters of adjacent nucleus that define the furrow. In Chytrids, it might be directly the interaction of the centrosome with the plasma membrane. But we wonder is, since centrosomes seems to be important to this process, are microtubules important? So to test this, we use nocorasol, which depolymerizes microtubules. And the idea is if microtubules are important and you treat with nocorasol, you would expect that the cellularization is inhibited or completely dysfunctional. But we were surprised to see is that on the top, you see a control cell. You have the cellularization, typical cellularization with very symmetric and geometric cellularization polygons. And on the bottom, you see the nocorasol treatment. And we were surprised to see that it's still capable of undergoing cellularization despite the fact that there is treatment with nocorasol. But what you can see is a very clear patterning defect. The polyhedra are disorganized, they're not as well formed, and they look actually floppy. There is a problem with the assembly of the lattice. To measure that better, I did a similar approach than before. I fitted ellipses into the sporangium, in this case, concentric ellipses. And what you can do is measure the intensity of fluorescence along the perimeter of each ellipse. That can give you an idea of the places where there is membrane. And you can calculate the peak of those fluorescent intensities and calculate the distance between peaks. And that can give you an approximation of the size of each of those cells that you have inside the sporangium. And if you see towards the last row of cells that are close to the plasma membrane, most of the furrows are perpendicular to the perimeter of the most outward ellipse. And that allows you to do the most accurate measurement of the size of those cells. So if you focus on the most outer perimeter of the sporangium, what we found is that the diameter of those cells is much smaller than the controlled cells. But what's more significant is that there is a high variability in the sizes of those cells, suggesting that even if microtubules are not necessary per se for the cellularization process, the patterning defect is because of a high variability in the distribution of sizes, suggesting that microtubules are important for the accuracy of cellularization. So we have that the selection of the division site in chytrids might be linked to the interaction of the centrosome with the plasma membrane. And this process does not require microtubules, but microtubules are important for the accuracy of the distances and the shapes of the cellularization lattice. Now, some of you might recognize this kind of process of the interaction with the centrosome with the plasma membrane, because it's pretty much what you see in animal intracellular cellularigenesis, in which the mother centriole recruits a vesicle, which is called the ciliary vesicle, and that vesicle fuses with the plasma membrane and allows the extension outside of the cell of the axoneme of the cilium. So part of my hypothesis is that both the docking of the centrosome during the cellularization in chytrids, because they also undergo cellulogenesis, is homologous to intracellular cellulogenesis of animals. Now, next, how is that actual three-dimensional lattice assembled? First, one of the most striking features of a Chytrid cellularization is that it has a lot of membrane compared to what is available in the plasma membrane. So to test what are the sources of plasma membrane in Chytrid cellularization, we use Brefeldin A, which is an inhibitor of trafficking between the ER and the Golgi. And across eukaryotes, Golgi is the main source of membrane for cytokinesis. So we wondered, is Golgi also the source of membrane for Chytrid cellularization? So when we applied Brefeldin, what we saw is a very drastic phenotype. And I'll play you videos of control cells undergoing cellularization on the left. And you'll see the typical assembly of the lattice that you have seen so far. And you can actually see the cilia at the end and the completion of the cellularization. But I want you to see two examples of the phenotypes when we treat these cells with Brefeldin. They divide, they go into the cortex, and they get stuck there. And they have this hypertrophy of microtubules that extend, maybe trying to start cellularization, and afterwards they die. This is another example in which it was treated in a later stage. They were already starting cellularization, and what we see is complete disassembly of the cellularization lattice, in which they collapse to each other. And the green things that you see there is just the cilia collapsing into each other too. The way we can image this a little bit more easy is that the same kind of sporangium you saw before, now you can stretch it out, and before cellularization, it looks like the one that I'm highlighting here. There's on the top is the plasma membrane, and as it progresses downwards, you see the formation of the four initials, those little vesicles. Those vesicles project inwards, branch, and merge. And eventually you have the beautiful lattice and then completion of cellularization. Now, what we treat with Brefeldin on the left, you see the typical evolution of that cellularization. But if you treat the cells early, when you have just the little vesicles being formed, they try to cellularize, but they get stuck very early on. They don't have any membrane to continue cellularizing. If you treat a little bit more advance, when they're starting to assemble the lattice, this network actually collapses into each other. And you can see the difference a little bit more clearly here if I zoom in. Typical assembly, then fail to progress, the phenotype that I call fail to progress, they just get stuck in early furrow initiation, or network collapse. If they have already started to cellularize, then they just collapse into each other because they have no more membrane available. And this is the quantification. Most of them, right after 40 minutes, pretty much they die. But they separated in these two main phenotypes. Either they collapse or they fail to progress, suggesting that most of the membrane of the cellularization comes from the Golgi, which is a conserved feature across eukaryotes. Now, we have membrane from the Golgi. How do they actually assemble the lattice? So something that is particular about the lineage of the opisthokonts is that they have myosin 2. Myosin 2 was an innovation of the lineage because it can dimerize with anti-parallel assembly. So the heads of the myosin are facing opposite directions. So when they interact with actin, they allow actin filaments to slide along each other. This sliding of filaments generate contraction or contractile structures. These contractile structures are what allow in animals the bending and folding of the tissue that defines morphogenesis, and the contraction of the cytokinetic ring, which allows cytokinesis in the way that we know in opisthokonts. So we wonder, is actin and myosin present in the cellularization lattice? So here you see two strains, the top one that is expressing LIFACT, which is a probe of F-actin, and in the bottom, a fluorescently tagged myosin regulatory chain, which is a probe for myosin 2 activity. And what you can see in both cases, is that both actin and myosin colocalize with membrane on peak cellularization, suggesting that maybe contractile actomyosin is important for the assembly of the cellularization lattice. Now, to test if actin was important, not only present there, but it was important for the assembly, I used latronculin B, which binds to monomers of actin and inhibits the polymerization of new actin filaments. You have here a control cell, which is undergoing cellularization. And because the cellularization patterns that we usually see are so stochastic, there's not a particular order in which the assembly is happening. I use a general measure of complexity to try to quantify them. And this is a measure of texture. The more complex the architecture of the membrane inside, in this case of actin inside, there's a higher texture. And you can measure this as homogeneity. Something that is very complex with high texture has low homogeneity. Something that is very homogeneous and is not very variable in its intensity of fluorescence, it's very high in homogeneity. So you can measure here in the case on the right, the control is ethanol treated. We have a relatively homogeneous texture. So you have the purple, which has a slight increase in homogeneity because it's increasing in the cellularization complexity. But when we treat it with latronculin B, immediately after we treat it, all the actin pretty much disappears, or the actin structures disappear. This is reflected by a high homogeneity. And we found is that right after treatment, we wash the latronculin B, and within 5 minutes, you have again started assembly of the cellularization lattice. This suggests that a lot of the actin that is being used for the assembly is under rapid turnover. You're polymerizing new filaments and depolymerizing them. And this is in agreement with a complex structure that is rapidly changing. So this is actin. What about myosin 2? So similarly, we have control cells here in the example. Green is actin and magenta is the membrane. You see that within 1 hour, the assembly of the complex lattice. But when we treat with blebistatin, which inhibits myosin 2, what we found is that they fail to advance. The sporangios just barely try to start cellularizing, but they do not progress beyond the early furrows or just the furrow initials. And they get stuck there, they don't progress. So this suggests that both actin polymerization and myosin 2 activity are necessary for the assembly of the lattice. If both actin and myosin 2 are important, this suggests that maybe contractile actomyosin is important in the assembly of the cellularization lattice. If the structures are contractile, they are under tension. So we wanted to test are these structures in early cellularization under tension. You can test tension in a system using laser ablation. If you cut the structures and they're under tension, they're going to recoil because they're dissipating the tension. So here you see an sporangium that is expressing the probe of actin, LifeAct, and we're going to target a laser to the place where I have my cursor. And if they're under tension, they're going to recoil. And as you can see, as soon as you shoot the laser, you cut the structures, there's going to recoil response in the actin. And actually, there is a response in the sporangium that tries to contract again and kind of repair the structure a little bit. So this suggests that maybe they're under tension. Something that we were also observing is that when you shoot the laser, the recoil response is dissipated through a lot of actin structures. And it was hard to track all the different directions in which this recoil was happening, and every sporangium is different. So again, I had to find a way to measure all this architecture, changes in architecture at the same time. So I use a trick that they use in fluid dynamics to measure changes in directions and speeds on particles in a system, and this is particle image velocimetry. So what you can do is do cross-correlation between frames of a video and assume that each pixel is a particle, and you can find what is the changes in direction and in speed of each of those pixels. And on the right, you see the image of particle image velocimetry of 1 second after the laser ablation. And in black, you see hopefully the direction of the vectors in which the particles are moving. And in color, the warmer colors are higher speeds. So you see two main groups of vectors, and those directions agree with the recoil response away from the ablation site. And you can see a video of this process before ablation, blue mostly. Right after the recoil, you have the rapid response away from the ablation site, and they come back again into mostly blue. So we did this on different cells and different days. And what you can see is that if you plot the top 10 velocity vectors at each frame, you see as we approach into laser ablation, you have a peak of velocity vectors, which it would be a representative of the recoil response 1 second after the ablation. And then almost 1 second right after that, they come back to pre-ablation conditions, suggesting that the tension in the system has been freed within 1 second. So this is early cellularization. There is contractile structures driven by actomyosin. Is actomyosin also important for the assembly of the action polygons that you see in late cellularization? So we did a similar approach. We targeted the laser to that bright dot in the center that I'm focusing with the pointer. What you can see right after laser ablation, that edge disappears and the polygons that are around dilate, suggesting that all these structures inside in the late cellularization are also under tension. You can measure this with a chemograph. So this bright dot on the left is the bright dot that we targeted with the laser ablation. And as we move towards the right, we are measuring the intensity profile of the pixels along a line which is parallel to the edge. What you can see as time progresses, we have laser ablation, and the tips of those edges that are generated by the laser ablation recoil away from each other. As we progress further in time, those tips start to gain brought back actually together by the sporangium, suggesting that there is an attempt of the cell to repair that hole that we made. So together with what we have is a working model of the cellularization in Chytrid fungi. And what we found is that Chytrids use a new mechanism to undergo three-dimensional cellularization, in which the old machinery of actomycin that is used both by animals and yeast is deployed in a very different way to solve the problem of three-dimensional cellularization. We have some traits that are shared with some of the two-dimensional cellularization programs in animals, which is, for example, migration of the nuclei towards the plasma membrane. And that allows the selection of the furrow sites, because that's another trait that is shared across animals and fungi, that the cytokinetic furrows is always assembled first at the plasma membrane, and then it moves inwards, what is called centripetal cytokinesis, in which the furrows start outside and they move towards the inside of the cell. And that's a trait that is conserved across model systems in fungi and in animals. What is different about Chytrids is that they don't use the typical two-dimensional rings that we use, that are used by model yeasts and model animals. They're actually relying on a strategy that might be homologous to intracellular animal cellularizations, in which the centrosome is docked to the plasma membrane, and that vesicle is what forms the individual furrows that are then extended by actomycin, and they merge and branch to generate the lattice, the three-dimensional lattice, which still we don't understand how they're capable to do this. What is important is that microtubules do not seem to be important for the early stages of the cellularization, and this is very different from what we know both in animal and yeast model systems, but it is important for the later stages for establishing the distances between nuclei and the correct shape of the polyhedra. So with that, I'd like to thank all the members of the Fritz-Leiling Lab and Dr. Mary Elting for helping us with setting up all the laser ablations, all our funding agencies. And I'm happy to take any questions that you have. Thank you.
Wonderful. Thank you so much. That was a wonderful presentation. Yes, thank you. I will open it up for questions, though I suspect that many students may save their questions for a conversation later, but I will open it up for questions in case anyone's got something they want to chime in with right now. Otherwise, I may ask some further questions. I'll give people also a second in case they have anything that comes to mind. Thanks. That was such cool and exciting work and amazing kind of images and techniques that you're developing. I was wondering, so like all the work that you're doing currently, you're working in sort of a single Chytrid species, I think, is that right? And I think a lot of kind of previous work has gone into kind of developing up to make kind of a tractable system to work with. Can you tell us a little bit about maybe sort of actually kind of doing this kind of work in Chytrids and what went into being able to build up kind of some of these, reporters and all these tools?
Yeah, so yes, you're correct. This is so far only in one species, Specialomyces punctatus, which is the model system that I developed in grad school, because up until maybe a month ago, it was the only genetically tractable Chytrid. So I've been fascinated with this developmental programs in Chytrids since my undergrad. So I've been working on Chytrids to try to build them up as model systems since then. And it was only in grad school I started doing a lot of computational work and evolutionary work, because there were no tools available to actually test many of the questions that were out there, at least at the mechanistic level. It's a beautiful long history in Chytrids of being used for tackling important biological processes. What was great about Chytrids in a way is that there's a lot of literature with beautiful experiments that allows you to have a primer of potential questions, but there were no toolkits to test them at the mechanistic level. So that's where during grad school, at the same time that I was doing a lot of computational work on evolution of cell cycle control, I had to start developing some tools to actually test them. So it was kind of going on both ends at the same time. And as soon as one thing worked, I started working with that one. And that was in this Chytrid speciomesis. Yeah.
Well, do you suspect that as maybe tools get developed across Chytrids, do you think there are these are mostly conserved sort of processes, or do you think we're going to see some sort of diverged functions?
So Chytrids are such a diverse group that I wouldn't be surprised that some details are different. But what we know so far from the classic literature in Chytrids is that even on the blastose, there's two main groups of zoosporic fungi, the Chytrid sensu stricto, which is Chytriomycota, and the blastocladiomycota, which are like siblings to the rest of the fungi. And Chytrids are outside. There's been changes recently, but they're siblings to blastose and the rest of the fungi.
Okay.
So even within Alleomyces, which is a model system within the blastocladiomycota, there's some very nice work with electron microscopy and some dyes that suggest some of the steps that I have found are also present in blastose. And all my findings agree with their findings. They suggest that nuclei are close to the plasma membrane, that it starts from the outside in. So there is some conserved features. So I think that in general, the principles are conserved across zoosporic fungi. There might be some details that differ, but I think I haven't arrived to a mechanistic level in which I can find where those details are different. Yeah.
Cool. Sounds like you've got like a big, exciting field ahead of you. So that's very cool. I love
We
that.
need more people working on Chytrids. So.
Yeah. Well, maybe you can recruit some. Okay. Well, I think that's great. If there's no other student questions, we will save them for the next session. Yeah, so I think please just join me all in thanking Edgar for a fabulous presentation. Thank you.
Thank you so much.
Thank you, Rebecca. So I'll stop the recording now, give you maybe until 11:50 to join back, and then Anthony will lead the discussion.
Awesome.
Thank you. Thank you, Rebecca.
Thank you so much.
Great. Thank you.